get cooking, let me do a quick public service announcement here. I'm the one of the advisors for a student club called the CIMA Club, which is the pre-med club for students interested in becoming doctors. And the CIMA Club and some other people are sponsoring it's a health fair event, and on June 2nd, which is a Saturday, I believe, over the summer break, obviously. And it's going to involve, amongst other things, some free healthcare checkups. You can get your blood pressure tested, and I'm not sure what else they're going to be doing there, but various health-related things. But also a 5K run, and if you're interested in any of those, let me encourage you to come on out to the health fair event. I will be there. I'm not sure if I'm going to actually run in the 5K. That might be a little embarrassing these days, but I'll be there doing something. All right, well, let's see. So today's lab is going to be a continuation of our cloning experiment on the green fluorescent protein gene from the jellyfish, or the octopus. Jellyfish, I think. But before we get into the actual cloning experiment, I want to start in on the DNA technology lecture. The actual hands-on lab work you do today is fairly short, probably less than an hour, and this introductory talk may take, I don't know, 40 minutes or so. All right, let's see now. But before we get into the sort of nuts and bolts of DNA technology and gene cloning, I was going to say, let me just introduce the subject. There we go. So a lot of this course is centered on the DNA molecule, and you know by now that it's the central molecule of biology, meaning that it really controls all the traits of any organism. The DNA has the genes, which are the recipes for the proteins, and then the proteins, like the enzymes for instance, carry out the tasks and give the organism its traits. So in the final analysis, it is your genes that give you all of the traits. And when they first started learning about DNA, the methods they used had for isolating it and analyzing it and manipulating it were fairly crude, but as time has gone on and technology has advanced, it's getting easier and easier to do all those things. I mean, just think about the last laboratory we did. Just, you know, in an hour and a half, you were able to isolate genomic DNA. And the techniques for analyzing it, meaning finding the sequence of the G's and A's and the T's and C's of any gene, is getting easier and easier. My thesis research was cloning and sequencing part of a gene in mice, the mouse growth hormone receptor, and you know that took years of work at the time. That was in the 90s, and now that would be trivial. Someone could probably do my thesis in a week or a month or something like that. Kind of embarrassing now that I think about it. It's not just analyzing the sequences of genes. We can now very easily change the sequence of them. There are methods of genetic engineering where they can go in and alter the DNA sequence, which of course then gives you a different protein sequence. You can modify the proteins by tweaking the genes, so to speak. And then you can go beyond that and move a gene from one organism to another. I think she's actually injecting DNA into cells from a different species. And I think I've shown you some of these examples already, but they're interesting. These are bacteria, and bacteria were some of the first organisms that were made transgenic. In other words, where they took DNA from one species and put it inside the bacteria. And this is just one example I thought was kind of interesting. This is crude oil, like petroleum from out of the ground. And occasionally you hear about these super tankers running aground and there's a big oil spill and it's horrible for the environment. And people have developed various ways of cleaning up spilled oil. Well, what this researcher here thought of is maybe getting bacteria involved in the process. And so he modified the genes of some bacteria to get them so they made digestive enzymes that could digest crude oil. And so he simulated a little oil spill here And then in this one, he added some of these bacteria. You can see that it's digested a lot of the oil away. So this was his idea for cleaning up oil spills. I had not heard that they're using this yet, because I imagine there's some danger with releasing bacteria like this into the wild, but it's a very interesting idea, certainly. Where they've actually applied a lot of this genetic engineering is to plants. I showed you this one. Someone took the gene from fireflies that allows them to glow in the dark and put it inside this plant, and you end up with a glow in the dark plant, which is fascinating but doesn't really help anyone. But for actual increasing food production, they've made a lot of progress. There's a big agricultural biotech company called Monsanto. I think they're from Missouri. And they've been at the forefront of genetic engineering a lot of crop plants. I forget which one this is. It might be a soy plant. But they put inside of the plant a gene for insecticide, for resistance from being eaten by insects. And so any insects that land on this plant and eat it are going to poison themselves. Now this insecticide doesn't harm human beings, so it would be okay for us to eat these plants. They call this the BT gene, whereas here's the control that didn't get that gene, and you can see the insects practically ate that plant alive. So this allows farmers to get a bigger crop yield, but the farmers don't have to spray their crops with insecticides to get it. And there are problems with spraying the field because the insecticides wash off the field into rivers and lakes and end up poisoning a lot of the good insects in the environment. This allows you to only control the insects that are actually eating your crops. Here's another example. I think this is a soy plant also. What farmers have to sometimes do is spray the field with herbicides to kill off weeds, because the weeds that are growing in the field compete with the plants for the fertilizers and the nutrients in the soil. But these herbicides don't only kill the weeds, they actually can hurt the crop plants themselves. And so this Monsanto company has, through genetic engineering, given the crop plants resistance to the herbicides, and so you can spray the fields and not reduce your crop yield. And here's what I also thought was kind of interesting. Certain plants can grow in salty environments, like when you drive, I don't know, past Elkhorn Slough, you can see various types of grasses and things that are growing in those salty waters. So those plants have salt resistance, but of course regular crop plants couldn't grow in that environment. Well, these researchers here analyzed the genes that give these plants salt resistance, and they transferred some of the genes from the salt-resistant plants into regular old crop plants, with the idea that you could grow plants in soils that have become too salty for regular crop plants. And this is another instance where I haven't heard of this one actually being used on a large scale yet, but it certainly holds potential. And it's not just plants. They can put genes from different species into animals. I've showed you this one. They put a gene for a human blood clotting protein that some people need as a pharmaceutical drug because they're missing that protein. They put that gene in a goat, and when they do milk, the goat can purify the protein out of the milk, and it becomes a pharmaceutical drug. And the holy grail of genetic engineering is to actually be able to cure human diseases by changing the DNA inside a living person. And so far they haven't really been able to do this. The problem is we have trillions of cells, and there's different cell types. There's blood cells and liver cells and brain cells. And they don't really have a way of putting specific genes into specific cells in our bodies yet. There was one example where they were successful at it. This child here was born missing a certain gene needed by her immune system, and they were able to get some of her stem cells out of her bone marrow, put in the missing gene inside a test tube, and then transfer those cells back into her body, and she essentially cured the disease. But that was a very specialized case, and in general they've not been able to do that for human genetic diseases. And this has led some people to think, well, you know, why stop at just diseases? Why not take things that are just normal human traits, like intelligence and strength and whatnot, and try to make those even better. And so some people think eventually you'll be able to supercharge your baby. You'll be able to say, I want my baby to have 10 million IQ and super quick reactions and great immune system and blah, 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 which I guess would be a good thing. But if you've ever seen that movie called Gattaca, where they sort of suggest that if we start improving ourselves, the people who decide not to get improved are going to become below average, not because anything's going to happen to them, but the average person will get smarter and stronger and whatnot. So we may end up with sort of a two-level society of the haves and the have-nots at a genetic level. Anyway, interesting to think about. Yeah, Rolando?
Can they transfer genes between, say, for instance, prokaryotes and eukaryotes?
We take a prokaryotic gene and put it in.
Eukaryotic gene and put it into a prokaryote.
Oh yeah, definitely. Like they can take human genes and put them in bacteria, and then the bacteria will make those proteins, and then they purify those and use them as pharmaceutical drugs. Like I think the people who take insulin injections and growth hormone injections, those proteins are produced in bacteria now, whereas they used to have to get them from cadavers and things. Okay, so so if one is going to has a gene of interest and they want to sequence it and perhaps genetically engineer it, you need large amounts of that gene of interest. Just to be working with anything, it's best if you have large amounts of it. And this process is called gene cloning, means methods of preparing very large amounts of whatever gene of interest, whatever gene you're studying. And there's different methods of gene cloning, but all the methods involve making what's called recombinant DNA. And recombinant DNA just means one piece of DNA that's created by linking together DNA from two different sources, usually two different species. And I diagrammed this a little bit on the chalkboard during today's lecture, where I said that there's these plasmids, these little circular bits of DNA from bacteria, but what they can do now is place genes from another species, like genes from a human being, and join them into the plasmid. And so this would be a piece of recombinant DNA, the DNA from two different species. So what I'm mostly covering on today's lecture is methods of gene cloning. Okay, so to clone a gene, look at it this way. You would need to get a circular plasmid, for example, and cut it open. And you would also need to cut up bits. If this was a human gene, you would also need to cut up bits of the human chromosomes, because human chromosomes would be longer than the blackboard, and that would be too long to stuff in a plasmid. So you needed some way to dice up a human chromosome into small little pieces. And so one needs a way of cutting pieces of DNA as part of gene cloning. But then you also, once you have your cut open bits of DNA, you also need a way to ligate them, to glue them together to form one piece of recombinant DNA. And the tools for doing this obviously are not scissors and glue, but they are enzymes. The enzymes that act as scissors for cutting open a piece of DNA, like cutting open a plasmid or cutting up human chromosomes, are called restriction enzymes. And the enzymes, so those are the enzyme scissors, so to speak. And the enzyme glue for joining together two pieces of DNA is an enzyme called DNA ligase. Let's look at the restriction enzymes first. Let's see. Imagine this is a piece of DNA. Maybe it's part of a plasmid or part of a chromosome. something like that. And that's one strand. Of course, DNA is always double-stranded, so the complementary strand would be down here like this. Like that. And of course, if this was a real chromosome, it would go on for hundreds of millions of base pairs. Now, so restriction enzymes are enzymes that latch onto the DNA strand and they scan along it. And the restriction enzyme's job is to cut the DNA like scissors. But they don't just cut it anywhere. Restriction enzymes look for a specific sequence in the DNA called their restriction site, and that's the only spot that they will cut the DNA is when they find their particular restriction site. And there are many different restriction enzymes, and each one has its own particular sequence that it looks for where it's going to cut the DNA. And you don't have to know these, but there's one called BAMH1